bio. Okay, so next up we have Jeffrey Ladish. Jeffrey works in information security at Reserve, which is a cryptocurrency startup, and previously worked at Paradigm Academy. His talk is titled "Nature's Source Code is Vulnerable and Can't Be Patched." Please help me welcome Jeffrey.
Hey everyone.
1 second. Let me just get you some mic.
So this is a talk I gave at DEF CON this year for the Biohacking Village. So it's partially geared at people with some biology background or some information security background. Just a quick question, or like a general survey, like how many people have a degree in biology or have like studied biology formally? Okay, just to get a sense. Great, that's okay. I was actually expecting more people at the Biohacking Village to like have more biology background, and I asked that question like maybe 10% of the people raise their hand, which is kind of interesting. I guess it makes sense. It's like a hacker conference. So the thing I asked people at that conference was if they had all updated their phones before coming to the conference. DEF CON is really notorious for like, it's just a conference full of thousands and thousands of hackers. So like if you bring like vulnerable devices, like it's pretty likely that you'll get them hacked. But I mean, one of the things I tell people on my team at Reserve is like the number one thing you can do to keep yourself safe is like make sure that your software is up to date. Because essentially, you know, in information security and in general computer security, you're in this constant arms race between attackers and defenders. And so someone will write a piece of code, and then there'll be vulnerabilities in that code. There generally almost always are, and people will find them and exploit them. But as long as like the defenders are patching them as soon as they're found and updating the software, you're generally fine. And so this is kind of like how the internet works at all, and like how we can actually like overall have a society that's based on a ton of computing technology. But the awkward thing about life, like biological life generally, is that we just can't update things very easily. And that's essentially what this talk is about. Is that like I'm arguing that nature's source code is essentially vulnerable and we can't patch it.
That's the opposite one.
The opposite. Okay. So I'm going to talk about sort of generally like what kinds of vulnerabilities exist in biological systems. In particular, what evolutionary constraints exist, and what human advantages allow us to overcome those constraints. And then the second section I'll talk about what we can do about that. So ways we can learn from cybersecurity, ways we can invest more in defensive technologies, and then just generally what we can do to prepare for the future. So starting off with vulnerabilities in biological systems. There's kind of an interesting question here because in cybersecurity it's fairly obvious what vulnerable means. Basically you write a software for a particular purpose, and if someone finds a way to abuse that software system for a purpose it wasn't intended for, it can be considered vulnerable. Like biological systems weren't really intended for anything in particular. Like there wasn't some grand evolutionary purpose. So I think the frame here is something like, well, we have opinions or preferences about what we want biological systems to do. Like I prefer that in general human biological systems don't like get sick and die, and that like ecosystems don't all collapse immediately and like not be able to support life. So in the context of this, vulnerable basically just means like functions as we would prefer it functions. So my background is, my biology background, and what I studied in my undergraduate days, is evolutionary theory. And so I think it's a really useful frame to think about why biological systems are the way they are. And one really, I think, kind of astounding constraint that all evolutionary systems have is that basically all evolutionary innovations have to basically follow a sequence of mutation after mutation. So in order for any mutation to achieve fixation in the population, that is to be in 100% of the organisms in that species, it has to be adaptive. Like it has to confer some positive survival advantages. Now, like occasionally, genetic drift will get you to fixation, but this is actually really rare. I think probably like much less than like 0.01% or something of all mutations achieve fixation this way. So for the most part, every little change has to be adaptive. And so like, I mean, like, part of the reason why is that like you can't build on something if only like 50% of the population has it, or you might be able to build on it a little bit, but like in the long term, it's not very likely to work. An example of this is like the recurrent laryngeal nerve or the vagus nerve. It's like an often used example in evolutionary biology. So when we were all fish, it was like relatively trivial for this nerve to go around our heart. It wasn't really around, our heart was kind of in a different place and our head was in a different place. We didn't really have a neck to speak of, so this wasn't a big deal. But as we started to develop a neck and our head got like further away from our heart, this nerve had to go through this awkward like loop around that part of our heart. And like as vertebrate necks got longer and more ridiculous, I'm looking at you dinosaurs, this nerve had to take this like really, really ridiculous route. Now like a human engineer wouldn't make this mistake. They'd just be like, okay, like scrap that whole like going around the heart thing, just like move it, just move it to the other side. But if you think about it from an evolutionary perspective, this would be catastrophic if just like one day one of the organisms just had a disconnected nerve when it was formed, like it wouldn't work. And so you see all sorts of constraints like this in biological systems. Another one is that like, why is it when we're embryos we still have tails and gill slits? Like, it's kind of weird if you're like trying to grow a thing to grow a bunch of features that you don't need and then like get rid of them. But of course, like if you're thinking about biological systems or like in particular development as a like program that you have to slowly unfold, making modifications really early on in the program is a lot harder than making modifications later on in the developmental program. So it's like you wait till the thing's like much more mature and then you like tweak a few things. Like that's going to be a lot easier than if you try to like, you know, take a few blob of cells and like change something fundamental about how it works. I think it's also the reason why you like, you know, for the most part, most vertebrates have like 4 limbs. Because it's really hard to add a limb because you have to add that really early and then it's going to have a bunch of cascading effects down the line. There's a lot more like ways in which evolutionary constraint act, but in the interest of time, I'm going to talk about some ways that humans can sort of potentially do better than that. So one thing that we can do is, this may seem obvious, but we can like actually plan ahead and do top-down design. So which is to say that we can basically be like, okay, I want this system to look like this. I have this goal of these particular properties, and we can like try to like draw a plan and then implement that plan. Like molecular biologists in the room might be skeptical, like, can we really do that right now? And the answer is like, well, no. A lot of ways we can't do this yet. But I think the argument is more that we in general have this capability, and we are building more and more of this capability in biological systems. And so essentially, like, natural selection can't do this at all. We can do this a little bit, and we're only going to be able to do this more. An example of this is one of the big synthetic biology lab, I think it was Craig Venture's lab, basically tried to figure out the minimal amount of genes necessary for a bacteria to function. I forget exactly how many. It was like, does anyone remember the number of, it was like, I think between 20 or 30, or within that order of magnitude. But so they basically like eliminated everything that they thought they didn't need and saw if the bacteria could still survive, and then completely synthesized all of those genes from scratch. Like they didn't just like cut them out; they like actually just printed them out and put them in a bacteria, and it could replicate, which kind of like it's a proof of concept for a future where if you want to create an organism, you like figure out, you like have a template. You have a template of like minimal genes, and then you add on some plasmids, you add on some functionality, so that we can begin to actually do like modular design of things, and instead of just like making these like really annoying point mutations, like how we currently do genetic modification, we can just be like, I'm going to add this module, I'm going to add this module, I'm going to take this template, I'm going to take that template, and start to move towards an actual like engineering paradigm for biological systems.
Supposedly they worked it down to 382 genes.
380?
382.
Okay. Off by an order of magnitude, but you know.
Just one.
Yeah, just one.
Actually, the actually working copy was 473 genes.
Okay.
Yeah, wow, that's quite few.
Okay, so another example is that we can do arbitrary gene transfer between species. So, like, we've been able to do this for a really long time. You just, like, take some jellyfish genes, put it in a plant, and you're like, aha, I have a glowing plant, this is great. This does happen in nature. Like, we definitely see, like, viruses sometimes carry genes between organisms. We sometimes see, or bacteria all the time can, like, exchange individual pieces of DNA. But what you don't see is, like, intentional transfer of arbitrary genes. So, like, in theory, eventually we should be able to take any gene and put it in any organism. Now, like, we'll have to, like, you know, figure out how to, like, stitch things together and, like, figure out all the building blocks. But, like, this basically opens up like a really large scope of things where we can basically take any innovation that evolution has created and move it to another organism. And then I think the thing I was kind of referencing before is basically the vision of synthetic biology, which is that can we actually get our tool set good enough so that we can modularize a lot of the process? So, like, kind of ignore all of the, like, details, but, like, what this system is doing is called Cello. And the idea is basically, can you create basically, like, biological circuits where you specify some inputs and you specify some outputs, and then you let, like, the program basically figure out how to prevent these sequences from interfering with each other, such that the inputs will accurately map to the outputs. It's pretty cool. You, like, can specify this in the program and hit run, and it, like, compiles, and then will output some sequence information, and then you, like, have a little plasmid that you can go put in your bacteria. It's still pretty limited. Like, you can't specify all that many things. But I think this is, like, where the future of synthetic biology is likely to go, where eventually biological engineering starts to look a lot more like software engineering. And the tools that you have are a lot more powerful, and what individuals can do is a lot more powerful. Okay, well, we don't have permission to see this video, which is fine. So, in terms of large-scale genome changes, natural selection can only move, you know, mutation by mutation. Sometimes these mutations can be quite big. So, like, you can, for example, like, duplicate a chromosome. It's like, you know, so, like, those mutations can be big, but what they can't be is, like, really large and really precise. So, say, for example, you wanted to, like, replace every gene with, like, an arbitrary different gene, or you wanted to replace every kind of one base pair with a different kind of base pair. Natural selection could never do that because the intermediate steps wouldn't be functional. But some synthetic biologists with a project called Project Recode are trying to give cells resistance to viruses. And I don't mean, like, resistance to specific viruses. They're trying to make them resistant to essentially all viruses, which sounds ridiculous, but it is potentially plausible. And the way that they're trying to do this is altering the transcription process. So basically they're like, all right, so we have a bunch of codons. They code for, some of these codons are synonymous, like UUU and UUC are coding for the same amino acid. And this seems like redundancy is, like, something that we don't necessarily need, although it might be kind of important. What if we just basically said, like, all right, we're only going to let UUU code for this, and we're only going to let CUU code for this one, etc. You basically, like, eliminated all of the synonymous codons except for one, and then you altered the translation machinery such that the like, RNA molecules would basically reject all of the other ones. Now you have a virus that has, like, a bunch of these synonymous codons in its code, and now it, like, can't be translated correctly. And if this works, this in theory, like, makes whatever cell, whether it's a bacteria or some eukaryotic cell, a lot more resistant to viruses. And I think that this is potentially a really terrible idea, even though it's really cool. Because if you could do this successfully, now you've created something that's much more adaptive than what has naturally evolved. Basically, by using this kind of, like, a little bit OP synthetic biology tool, which is, like, these large-scale changes. And so the analogy here is antibiotic resistance. Like, if you just give something antibiotic resistance in an environment with lots of antibiotics, it's going to really outcompete these other things. And so if you give a arbitrary organism virus resistance in an environment where there are lots of viruses, which, spoilers, there's lots of viruses everywhere, you're going to give it a really big unnatural advantage. And so, like, this is just this is like a proven concept, but I think it really like illustrates the point that eventually, if not soon, human-engineered organisms will be able to exploit design constraints and naturally evolved systems. And so, essentially, all life is vulnerable, and we need to think about how we want to go forward with that. Okay, so those are some vulnerabilities, and now let's talk about what we can do about it. Alright, so I give this talk at DEF CON, it's a cybersecurity conference, and I also work in cybersecurity, so my frame is like, okay, there are some analogous things we can learn from cybersecurity. Let's see what those are. I talked about this a bit earlier, like, why does this work? How can the internet continue to function? How can like we actually use systems even though we hear all the time about how insecure they are? And we can update software. We have other kinds of defenses too. So like, we have software updates. We also have antivirus software. Antivirus software is essentially looking for patterns of code that we know to be malicious, and then flagging those patterns and freezing them or deleting them. I think another really important class of defenses in computer security is basically architecture updates. So you can like keep patching something and keep like mitigating vulnerabilities, but like at some point you want to stop using XP. Like even if you're on Service Pack 3, like just like don't use XP anymore. XP is vulnerable. Like we've just given up on that one. And partially it wasn't designed to be as secure. We didn't know how to design it to be as secure. So like as we make new software and as we make new operating systems, we like change fundamentally how they're designed so that they're more secure. And I think in a lot of cases, like we have actually seen really big improvements. Like if you look at like a modern Mac or a modern iPhone or even a modern Windows computer, they're a lot more secure than their analogs were 20 years ago or 10 years ago, because we've actually learned a bunch of stuff. All right, so biological systems, we can't do software updates. Maybe eventually we'll be able to do that, but we're not anywhere close. Like, if I say I'm gonna like modify how your immune system works and like give you new kinds of T cells or new kinds of B cells, like run away, like don't listen to me. We have adaptive immunity, so we do have something like an antivirus program. Like, obviously that's where it got its name. It's like we have an adaptive immune system, which is great, and it can be trained and we can use vaccines. But we can't do architecture upgrades. Like, we couldn't do them in a single lifetime because you have to change a big part of how the design worked. But given that we can't even do software updates, like architecture upgrades are like software updates, but just for the whole thing. So antivirus programs are doing this thing where they receive automatic updates, basically, and like how this works behind the scenes is like some security researchers are going through and like looking at the vulnerabilities and then like basically, you know, putting in what the specific patterns are, and then your antivirus program is grabbing those patterns from the internet constantly, and then scanning your system. These like antivirus programs don't work very well if you stop getting updates, because they'll only be able to look for patterns that they've seen before, and because exploits are like constantly innovating, you're not going to catch new ones. Okay, so in some ways our adaptive immune system is better than antivirus programs because they can learn on their own. So they're not just reliant upon an internet connection to get new updates, which is cool and like really amazing when you think about it. But the updating system is, not in terms of learning, but in terms of how humans can update the adaptive immune system. It's really slow and annoying and expensive because you have to like actually be poked with needles. Like if you thought about if every time you had to get your computer patched, you had to like stick a needle or like go to the store and buy a needle and like stick a needle in your computer. That would be really annoying. You'd probably get a lot more viruses. And so like this being slow is kind of annoying. And I think this third point is kind of unfortunate, which is that we have some proof of concepts that organisms can be designed or like can evolve to circumvent the entire system. So with HIV does a very effective job at just circumventing the immune system. So we have this awesome like antivirus system, and then sometimes like there's an exploit, and the exploit can't be fixed by the immune system. Like there probably are antigens that can cause the immune system to do a better job fighting HIV, we've discovered some of these, we're working on a vaccine. But the fact that HIV has basically resisted our efforts to develop a vaccine for it for decades speaks something to how difficult this task is. And given that this is just a naturally evolved virus, if the claim is true that we can design much better systems than some biological systems, especially at the small level, it may be the case that there are lots of immune vulnerabilities that aren't patchable that we could discover. And I think this is the real risk. It's not just that HIV might not be patchable, it's that HIV represents a class of vulnerabilities that might exist. So, I think whenever we're talking about really powerful technologies and the future development of these technologies, it's really important to think about differential technological development. Basically, like, technological development doesn't happen like across the board equally. Our roads and bridges haven't gotten particularly better in the last few decades, while our computers and our biological systems have gotten, like, our biotech in general has gotten a lot more powerful. So in theory, we have some ability to like try to choose where we invest in terms of technological development, and ideally we favor defensive technologies over offensive ones. Unfortunately, I think most trends in biotech today are favoring offense over defense. So like DNA synthesis becoming a lot cheaper and a lot easier. Well, it definitely has some benefits to defenders as well as attackers. Like, you could potentially use this, or DNA synthesis, RNA synthesis, to make vaccines a lot cheaper, or make them a lot faster. So you might be able to like respond at hospitals and actually produce vaccines there. CRISPR, of course, is like making gene editing a lot easier and a lot cheaper. But I think that this is like overall going to benefit attackers more. And then just in general, I think lab automation technology makes it so that many, many more people can do powerful things with biology. And all else equal, I expect that kind of to benefit attackers. So there's a question of like how we can tilt the balance towards defense. I have some ideas here. I'd be really curious in general to hear yours. You probably have some better ideas than I do. I haven't looked at this too much, so I think in general where we can increase detection, that's really great. Like, I don't know if we can eventually get like way better monitoring at airports or other like choke points where you see a lot of people passing between places. I think there's something really tricky where it's like we are now looking at vulnerabilities that could affect whole ecosystems, and I don't know how you would do monitoring at the ecosystem level, but it'd be really good if we could, right? Like, would we know today if someone was using a gene drive to like wipe out an insect population? Well, like, probably not. If it was an insect population that no one cared about, like, that would just go unnoticed. Now, I don't think anyone's doing that, but like, I do think that's the kind of thing that would be good if we could know. I also think that we just really need better threat modeling. Like, I see a lot of research going into like the threats that we already know about, like bioterrorism with known agents, and like potential bioweapons with known agents. And I'm like, okay, but like biotech isn't slowing down, and like I think we need to be more imaginative in terms of what we expect the future of biotech to look like and what we expect the future of biosecurity to look like, and develop accurate, at least it's hard to develop accurate threat models when there's so much uncertainty, but at least like come up with a better range of threat models to encompass the different situations we might find ourselves in. So this is Bruce Schneier. He's like a pretty famous security expert. He talks a lot about the security mindset, which is essentially imagining yourself as the attacker and imagining all the possible ways that you could break a system. And I think that we essentially need to apply this to the biosecurity world and like really act with a biosecurity mindset. And just, I mean, I'm kind of preaching to the choir here because we're all kind of here to do this. At DEF CON, I was like, all right, we're like putting implants in ourselves and we're like hacking medical devices, but like also we have to think about how people can use this, like some of these things for harm. And this is a question of like, all right, what does the future of biotech look like? Is it dragons? I hope it's dragons. I hope it's not global pandemics that kill everyone. But I do think that like you need to think about potentially how powerful this technology might be and like what reference class is it in. And I don't think this is at all certain. Like, I think that it could be as powerful as, like, we could be looking at technology that becomes more impactful than nuclear weapons. Like we could be looking at weapons that that really change the strategic balance, or maybe these applications turn out to be really hard, and instead we're looking at more of the reference class of sort of global pandemics in the past century, which I think is a lot more manageable and requires a lot less effort and resources. And so I think that we should try to develop better models of what the future might look like, and figure out how to make drones. So I think that the biohacking communities are really interesting, and one thing is that they're full of really weird people, which I think is great. This is probably also true of rationalists and EAs and other groups like that. And I think that this is actually a big advantage, because a lot of people are going to be pretty constrained in what they can think about and what possibilities they can imagine. But if you're willing to think outside the box and willing to actually engage in weird speculation, I think you can potentially find some of the high leverage points. These are some bats wrapped in blankets. I studied bats in my undergraduate and I really like them. But it's not related to the rest of the talk. Except that bats carry a lot of diseases, which is unfortunate.
That was a really great talk. Thanks so much. I was curious, in your model, I thought it was really interesting how you were contrasting software engineering models with biological engineering, I guess. But what was the what was the nuance in terms of how you considered software versus architecture in biological systems? I don't think I completely caught that.
Yeah. Well, I think it's hard because in software system there is a really coherent concept of architecture, because the whole thing is modular from the very start.
Yeah.
And in biological systems, I think there are architecture analogs, but there's not any architecture, or there aren't many architecture analogs in how we're currently designing systems. Just because mostly what we're doing is modifying things that currently exist.
Right. Yeah.
In terms of I think if you looked at, you might be able to look at things how have cephalopods evolved nervous system compared to how vertebrates have evolved nervous systems, and look at other cases of convergent evolution and look at where there were architecture choices essentially that natural selection took, or looking at immune systems and looking at okay, are they fundamentally doing the same thing, or are they did they choose a different approach, and just trying to figure out places where there are potentially design trade-offs or design principles that you could infer.
Right. So architecture is the way the types of genes that you use to achieve something, whereas software in your model is how you actually do the transcription or translation.
Yeah, the implementation versus the high level principles. It's kind of weird to talk about natural selection having high level principles, but the analogy of that.
Yeah. I have more comments. Anyone else do? A couple of other things I wanted to. So one thing I wanted to point out, in your discussion of countermeasures against rogue engineered organisms, I thought that was very interesting. And I think so. Yeah, I'm not super confident about this, but I think there is actually a lot of pretty readily translatable kind of infrastructure for improved detection. So DNA sequencing, for instance, is vastly cheaper than DNA synthesis right now, and I think it could be ported over for more biosecurity technologies, and also things I imagine that if you want one interesting way you can find out if something was synthetically engineered was to see if there are specific barcodes in your organism that relate to whatever cloning method you use. So if you use Golden Gate for DNA cloning, it all sometimes just leaves very specific barcodes. So I think stuff like that are things that you can potentially do.
Okay. Yeah.
Measures. I'm not 100% sure someone is working on that, but I know IRBAR runs similar types of programs where they try to do detection.
Yeah, that's a good point. Especially that sequencing in general should favor defense.
Yeah.
Because it's you can get more knowledge with sequencing, but it doesn't allow you to build more things.
Yeah, but I think it is true that there's no direct way to tell if something has been genetically modified and probably invested in it more.
So what you're saying is that you'd be able to tell whether a pathogen or an organism in the wild was genetically modified versus not?
So I don't know if I'd be that confident, but I'm saying one way you could potentially do it is if you look at some of the DNA specific sequences that often get inserted into a genome when you do certain types of cloning methods, that might be seeing if organisms have that barcode might be one way to tell. I guess what that would come with caveats how frequently do you find those types of sequences, just like generally in organisms at all, versus like how specific is it to like DNA cloning methods? But like if I had to like guess on a way we could improve countermeasures, that would be one. Yeah.
Cool.
Yeah, to go on that, I know that there are some people who are working on trying to create gene logging, so like creating a mechanism whereby a gene would like record an inert, would insert into itself an inert like gene that records like any changes that have happened to the rest of the genome. Yeah, it's like completely conceptual. It's not like anywhere close to being like ready. But I know some people are working on that, and if that ended up working out, like so you could sort of like hack ways of determining if a system is tamper proof.
What was it called? I very much like to look up what
I don't know. My coworker went to a different conference and came back and talked about it, so I don't know the name or have any links that I could try to find them.
It's like, it makes a certain kind of sense that it would work, because if you have something that has most variants of different sequences, then like whatever you use to cut it will also cut that. But
I look up the everything is about logging in the sense of cutting down trees.
Oh yeah.
Unfortunately.
yes. Cool. Thank you so much.